YTB is a novel autologous CAR T-cell product made by Novartis. It's been developed in phase 1 studies, and we presented the phase 2 data in diffuse large B-cell lymphoma. YTB it uses the same CAR, the same chimeric antigen receptor as tisagenlecleucel, otherwise known as Kymriah, but the manufacturing process has been significantly modified to deliver patients a T-cell product that has more fitness, the capacity to expand within the patient, because we've seen clear preclinical research that shows that those fit T-cells are the things that help control lymphomas. The manufacturing process is also very short, so reduced from a couple of weeks down to just 2 days. And so the goal with the YTB clinical trial is to deliver the CAR T-cells faster and to demonstrate that manufacturing is reliable with that sort of 2-day process. And this will help achieve improvement of 2 problems with CAR T-cells. One is that sometimes patients don't benefit from them because their CAR T-cells are exhausted. And so we hope to deal with that by providing a more stemness-like lymphocyte product. And the other is that some patients can't wait for too long. So we hope to improve that by reducing the overall turnaround time as all of these processes are optimized. So we treated around 40 patients with large B-cell lymphoma in this study, and we gave a dose of CAR T-cells that was about 1/20th of the dose of Kymriah. And the cells, instead of being expanded in the lab, expand in the patient's body, and they start from a lower point. But what happens is they expand up to as high or higher levels than Kymriah. And for the 40 patients that we treated, we saw roughly a 60% complete remission rate, which is significantly higher than the commercially available products at this stage of development. And we saw a very favourable toxicity profile, so similar to Kymriah, but later onset of toxicities, low rate of cytokine release syndrome, very low rate of neurotoxicity, but just a little later than we would expect to see with its predecessor Kymriah. And so we think this is enough to say that this is a promising product and that it's well tolerated. And so it might be an important option for patients. And it probably supports this hypothesis that having a more lymphocyte population with more stemness might improve responses. But obviously there's more work to be done.